he would be I asked him to give me a few first.
Okay. So are you going to then ask the questions, Colleen, or I should look at chat?
I think we decided that you would look at chat. I think it might be more seamless instead of going back and forth, if that's okay with you.
Sure. Okay. So then I will get out of screen share at the end and go back to chat and answer questions. Okay.
Yeah. So I just put one in right now, and then let's see if it works.
Yep. What is about Princeton ecosystem? Yep. Okay.
Right. I love it when a plan works.
Just you might be seeing at the top, it says live on streaming service. We're not live yet. We're controlling it on the other end.
Right.
So don't worry.
Yeah.
You sent that privately to Bob, correct?
I did.
Because I didn't see anything on that.
Right. Yeah. So because I think if I send it privately to Bob, then it doesn't go to
No, that's fine.
Okay. On the
Make sure.
Yeah.
And another way we can also chat with each other, and the way that I'll give everyone a time update is if you just select panelists in the chat down there in that little blue bar.
Oh, I guess yeah.
Because the other selection is panelists and attendees, although I'm actually going to disable their chat right now.
Okay.
And disable their raise hand.
And then, Bob, I will also say when it's last question, because I'll be watching the time. So I'll just say last question and then put the question in.
Okay, good.
And then when you wrap up that question, it'll come back to me and I will just close out the session.
Okay. Mm-hmm.
Unless we're running out of time, and then it'll close out on us anyway. Mid-sentence, it'll be done.
Will come out.
Comes out, right?
Yeah, exactly. So just a reminder, just to talk through to make sure I'm on the same page with you all. So at the top, you'll see the slideshow and the music. You've probably seen quite a few times now. And then when we're ready to start calling, I'll say stand by in the chat. And then as soon as you see that orange logo and the music stops, you can start speaking, and I'll put you in spotlight so everyone can see you. And then again, you'll pass it to Pablo, and then Pablo, you'll pass it to Bob. Bob, you'll screen share. Don't forget to stop your screen share when you're done, because sometimes that's an easy thing to forget. And then we'll do our Q&A with both Bob and Pablo, and then you'll hand it back to Colleen.
Once I introduce Bob, do you want me to turn my camera off, or you'll simply, I can do whatever you prefer?
I would leave your camera on because I can cover who's being seen. Do mute yourself and unmute yourself as needed, just in case something happens in the background where you are. But leave your camera on because I can control what the audience sees from there.
All right, very good.
Great. And Colleen, you're monitoring the Pathable chat, right? Are you okay with your setup to see that?
No, because I'm asking them to put it in polls, right? And then that comes.
Yes, polls. That's what I meant to say, exactly.
Yeah, and then at the bottom of my screen, I can look at polls, and it should be in there, correct?
Yes, on the Pathable page.
No, wait. On what?
So it's separate than the Zoom chat. So the Zoom chat is just for us, and if you want to see what people are writing in Pathable, you need to have that open on your browser as well. And I can send you the link if you want to do the easy way to see that.
That would be great.
I've got it open too, Colleen.
Wonderful.
I'll back it up.
Oh, yeah, actually, Tracy, that would be great. So I've got 3 questions that I'll put in. But Tracy, if you're monitoring it, if you're seeing questions, would you just put it in the chat to Bob? Yep. Great, thank you.
So I see the question on my chat, and I just keep this chat open, and this is where the questions are going to be relayed. Okay.
Yeah.
For the Zoom chat, exactly.
Great. All right. So we'll tag team. I'll get the first 3 questions out.
Got it.
Excellent.
Marvelous. Thank you.
So Pablo, how many times have you had to go through this procedure and this engage?
No, I have 3 appearances, so I've done three trainings.
Got it.
I'm starting to understand it.
We're just I was talking with Tracy this morning, or communicating with Tracy this morning of like just back up because I'm like my biggest fear are the technical glitches of suddenly my computer will go down or something, and so Tracy's here to pick up if something goes awry.
That's great. And internet is we just can't control it, unfortunately. Even great internet sometimes is not great. It's like magic, black magic.
Yeah. Normally mine's really pretty stable, but the last couple of days, I don't know if it's just because I've been doing so much with Pathable, but there are a couple of times I've been kicked out of Pathable, and it's again.
That's no good.
Queen, I'm not normally a superstitious person, but if you could change topics, that would be.
Okay, sorry.
I did want to say,
I'm-
oh, Bob, it looks like you were, I was just going to ask you if you could actually tilt your camera down just so there's a little less headspace above you. We'll get you a little more straight on there.
There, yeah. And
Yeah.
move it
That
back
looks good.
to get, okay, or I guess forward to get a little, okay. How about
Very
Is that a
nice.
virtual background or is that
That's our dining room. And
okay?
there's a picture between the two lamps I've taken off the wall, but that's our, and this is the most neutral of backgrounds in our house. I tried putting up a sheet in the basement and that just looked a little too hokey. So this is the best, this is what we'll do.
You could have put the sheet up and say you're inventing in your garage or something, being
Right.
really entrepreneurial.
So many garage stories.
There's actually like a lot of authenticity and warmth in all these people like coming in from all these different locations, virtual backgrounds, actual homes. It's
Yeah.
kind of a nice touch, actually.
Yeah.
Humanizes
agree.
everyone.
And I put in the chat before the session started just information about the audio and then reminding them about the polls. But I'm going to remind them again when I come on screen.
Yeah, I see those there. That's great.
Okay.
Okay.
We're trying to do that at the. Tracy asked all the session leaders to do it at the beginning of their sessions.
That's a great idea.
And the engineering
Welcome, and thank you for joining us for the special presentation of the Dean for Research Award for Distinguished Innovation. I am Colleen Burris, Director of Corporate Engagement and Foundation Relations and co-chair of Engage 2020. I have a few housekeeping announcements. The best way to watch the session is in the theater mode, so please click that. Also, please submit your questions in the polls tab, not the chat. Now I have the pleasure to introduce Princeton's Dean for Research, Pablo Di Benedetti, the class of 1950 professor in engineering and applied science and professor of chemical and biological engineering, who will present this award. Pablo?
Thank you so much, Colleen. Good afternoon. I'm delighted to welcome everyone to the presentation of the inaugural Dean for Research Award for Distinguished Innovation. I'm Pablo Di Benedetti, Princeton's Dean for Research. Princeton University's faculty and researchers have long played a really critical role in advancing the frontiers of knowledge through their groundbreaking work in the arts, humanities, social sciences, public and international affairs, engineering, and the natural sciences. However, the advances and innovations generated at Princeton have not always made their way through the university's gates and out into society at large. Princeton faculty and researchers are pioneers across an extraordinarily broad spectrum of human inquiry, and their breakthroughs often have the potential to make a tangible difference in everyday lives if these discoveries are more widely shared with the world. A few years ago, Princeton resolved to prioritize bringing the cutting-edge work being done on campus to the wider world, both now and into the future. This strengthened commitment to living the university's informal motto, in the nation's service and the service of humanity, has been taking shape all across campus in numerous and exciting ways, including within my own unit, the Office of the Dean for Research. Among our most notable endeavors is the newly created position of Vice Dean for Innovation, held so ably by who has been championing our increased emphasis on transitioning research into innovative projects that provide benefit to society. One of Rod's excellent ideas is our new Dean for Research Award for Distinguished Innovation, which recognizes explicitly the importance of impact and impact of innovation in society at large. This award supports Princeton's commitment to fostering a culture of innovation on campus and increases the visibility of our discoveries. Open to all Princeton faculty across all disciplines, the award acknowledges the vital importance of bringing creative university research to bear on the challenges facing our world and placing that research in the service of humanity. We are truly delighted to present the first annual award for Distinguished Innovation here at Engage 2020. An array of exciting innovations and innovators were nominated this year. The selection committee, comprised of external innovators and entrepreneurs, enthusiastically recommended that the inaugural award be presented to Professor Robert Prud'homme of the Chemical and Biological Engineering Department for the invention of flash nanoprecipitation, a technology with numerous applications, perhaps the most significant of which is improved delivery of therapeutic drugs throughout the body. The result of over 2 decades of research in Professor Prud'homme's lab, the flash nanoprecipitation process involves the rapid contacting of immiscible ingredients to create uniformly sized nanoparticles. The technique can be used to encapsulate drugs within tiny but protective biocompatible particles to improve their transport and effectiveness within the human body. This makes it possible to target precise locations such as tumors and then slowly release these drugs over time, greatly increasing their efficacy. Flash nanoprecipitation works with numerous drugs, including biologics, which otherwise would degrade quickly in the body. The method is relatively low cost as well as scalable and has additional applications such as in UVA filters for safer sunscreens, upconverting phosphorus for deep tissue medical imaging, and silver nanocolloids for antibacterial formulations. Robert Prud'homme has been a member of Princeton's faculty for over 40 years and served as a founding director of the program in engineering biology for much of that time. His research is aimed at understanding how weak forces at the molecular level, such as hydrogen bonds, determine macroscopic properties on larger length scales. Over the years, He has investigated complex fluids and soft matter systems, such as polymers, surfactant phases, vesicles, biopolymers, and emulsions. A talented problem solver, Bob has consistently found ways, Bob has consistently found ways to channel his curiosity towards the solution of practical problems, ranging from therapeutic drug delivery to the control of wax formation in crude oil pipelines. Throughout his career, he has had extensive research collaborations, both within and outside the university, including with corporations, national labs in the US and abroad, and scientists at the National Institutes of Health. He has written more than 200 publications and has over 25 patents to his credit. He is the scientific co-founder of Optimios Life Sciences, a startup company that is developing his nanoparticle enablers technologies to create improved delivery methods for medications targeting diabetes, autoimmune disorders, cancer, and diseases of the eye and the nervous system. In 2018, Bob was named the Research Council of New Jersey's Edison Patent Award winner for the initial flash nanoprecipitation patent. The technology has resulted in 30 additional patents and applications, many of them licensed by Princeton to major life science companies. I am truly delighted to present the inaugural Dean for Research Award for Distinguished Innovation to Professor Robert Prud'homme, whose hard work and creative thinking in the service of humanity led to the development of flash nanoprecipitation method as a solution to problems in drug delivery. Please join me in recognizing his accomplishments. And before we do that, Bob, I'd like to show you a beautiful award that we will present. I would have liked to give you this personally, but I promise it will be delivered to your office. Bob?
Fair enough. Okay. Well, thank you, Pablo. I mean, it's a tremendous honor to be conferred this award, and I deeply appreciate it, especially because it really addresses what I have a real passion for, and that is innovation and entrepreneurship. What I'll do today in this talk is very different than I would give at a Controlled Release Society or American Institute of Chemical Engineers meeting. What I'm going to do is take you, use this as an opportunity to say thank you to the collaborators that made this possible, because Princeton's an amazing place, and it really nurtures people who sit in ivory towers and think very deeply about hard problems. I mean, Andrew Wiles and the Fermat's theorem. At the same time, it's an environment that's enabled me to collaborate and form a network of relationships, collaborations inside the university, with other universities, and with corporations to really advance technology. And that's why I became an engineer, is to try to do things which translate basic science and engineering to make a difference in society. Let's begin. Let me share my screen. Here we go. Let me go to full screen mode here. Let me turn on my pen and proceed. So I've entitled this Nanomedicine from High Tech to Global Health. And I said, I'm going to give you vignettes or the history. And as Pablo said, this is history that goes back about 2 decades. And it would have been impossible without a tremendous cast of able grad students and postdocs. The short list is here, but also without funding support, which we have received from NSF, NIH, the Gates Foundation, and a series of other groups that have funded our research over this long period of time. I got into this again about 2 decades ago when I heard a talk from a vice president of Pfizer who came to Princeton and spoke, and he said that 40% of the new drug candidates coming through their pipeline were so hydrophobic that they couldn't be delivered easily. And so, go to a highlighter. And there was a need for new techniques to deliver these compounds. And whether you delivered them by what's called enhanced permeation retention, that is, if you can make a small protected nanoparticle in a cancer tumor, the vasculature is growing so quickly that there are defects in the vasculature. And these nanoparticles will leak out through the vascular defects, embed themselves in the tumor, and then release the drugs. So that's one thing that motivated our work. Or you might want to specifically target the drug. And by that, I mean, you might want to have a targeting moiety on the surface of that nanoparticle. The nanoparticle contains the drug itself, and you specifically target it to a cancer tumor. So the green dots here are nanoparticles that have been targeted to a breast cancer cell line. A big challenge is if you, unfortunately, are in the hospital with the cancer, need cancer therapy, you're not given one drug, you're given multiple drugs. And so the ability to deliver multiple drugs from a nanoparticle was something no one else was able to do when we began this. And then imaging. So it's not enough to say, I am going to target, or I think this drug goes to the cancer tumor. You need ways that you can image where things go. And then, in addition to small molecule drugs for oncology, there were opportunities, especially developed more recently, for delivery of proteins, peptides, and oligonucleotides like mRNA. So those were the challenges. And if you look at those challenges, it's not just making things small. You need to be able to control surface functionality. You need stoichiometric, which means equal encapsulation of different species, and you need imaging and targeting. And then, most importantly, you need something that can make an impact. And so these are two quotes that have really been important to me. The first is out of CNE News, and it says, over the next few years, some of the complex theranostic strategies published rampantly in chemistry journals will fall out of contention. For me, something that's too difficult to make or too complex to sustain in large-scale production is not what we're interested in. And that's exactly where I fall. And I've been a consultant with companies that had $25 million in startup. They had a science article to prove they could cure cancer. They then tried to make it at large scale and couldn't. They asked me to come in and help them, and I said, you're doomed. They continued until they went bankrupt and ran out of money. So the problem of making things so that you really can translate them and make a difference is important. Scott McNeil was the head of the National Cancer Institute's Nanotechnology Laboratory. He had this to say again in CNE News. Another big hurdle in developing nanomedicines is scaling up the synthesis of the particles. Developing a synthesis that yields particles on a consistent basis, this is still a difficult process. And so those are the kind of challenges and the kind of goals that I've set for myself and for our group. So we're going to begin this story. As I said, what I'm doing is, this is really an opportunity to give thanks to the people who really contributed to what we've been able to accomplish. This begins about 20 years ago with Brian Johnson. And Brian was a Merck researcher who was sent to get his PhD, sent back to Princeton and paid for by Merck. And the story here is a story of a failed PhD project. And it failed in this respect. Back then, there weren't any of us who were really doing pharmaceutical work on our faculty. Brian signed up to do his PhD with me and to look at a block copolymer. And Rhodia, a French chemical company, had recently come up with a new synthesis technique that they thought was going to be revolutionary and allow them to make lots of block copolymers. And they sponsored our research to look at the phase behavior and properties of these block copolymers they were going to make. And Brian signed up. However, it turned out that Rhodia couldn't make the polymers they thought they could, and they could only give us one polymer. So here you are with one polymer, and you can't really do phase diagrams and behavior with one polymer. About 2 years previously, I'd been invited by BASF, Dieter Horn, the head of their polymer physics group in Ludwigshafen, Germany, to come and visit the lab. And he had taken me through research that they were doing on rapid precipitations to make small particles. And at the time, Dieter asked me if I wanted to get involved and wanted to be sponsored. And I said, no, because it wasn't very interesting polymer science. What they were doing was making these particles and embedding them in Jello. They're essentially making fruit Jello. And that polymer science of that wasn't very interesting. But now, here a couple of years later, we have Brian with one polymer, and the polymer is a block copolymer. It has a hydrophobic group, shown in black there, and has a hydrophilic group, which makes it, that end wants to be water soluble. And I said, I bet we can do this rapid precipitation process and use this polymer to make stable nanoparticles. So this is the only real technical slide I'll give you to describe the process overall. And the process has two parts. The first part is to bring in our solute, our drug, and then this block copolymer, hydrophobic block, hydrophilic block, in a water miscible solvent. And then the key here is the mixing devices we have developed. And this is not microfluidics, it's controlled turbulence. I had learned about turbulent mixing in my early career. I had worked in that area, and Chris McCosco, who will come later in the story, was the one who really taught me about turbulent mixing. You mix that against the anti-solvent, non-solvent water. And so in this millisecond and a half, and that's the beautiful work Brian did, was to quantify these mixing kinetics. And so this mixing is over in a millisecond and a half. And now the hydrophobic drug starts diffusing and aggregating, making bigger and bigger clusters. The hydrophobic block of this polymer is landing down on the surface of these clusters and aggregating and forming this final nanoparticle that contains the drug in the core and these water-soluble polyethylene glycol chains on the outside. When enough of these have absorbed, that stops the aggregation. So the start is mixing, the stop is when the particles have a dense enough layer that they no longer aggregate. Now, part of the story here that'll come in later is if you have too much polymer, or if these aggregate too quickly, you form what's called a micelle. Now, micelle is just the same polymer with no drug in the core, but with the same chains on the outside. So this is what Brian developed in his thesis. And this is just an example from Brian's work. And what this is a molecule called beta carotene. And if you do this flash nanoprecipitation, rapid mixing, you make these beautiful 200 nanometer particles, which gives you greater bioavailability of this particular compound. If you use exactly the same chemistry, but don't do the rapid micromixing, you can see you make these 20 micron big platelets. So it looks like sort of a glitter fingernail polish. So this is what you want. You want these nanoparticles. If you don't understand how to do the mixing, you end up with these macroscopic crystals, which are not very useful. So that's what Brian did. As Pablo said, Brian's several publications, as well as the patent, became a foundational patent that are now, since 30 patents in this field come from his initial patent, which won the Edison Patent Award. Based on his work, we were able to submit an NSF proposal for a nanoscience research center, and which I led, but it brought in this wonderful cadre of colleagues. So from Chris McCosco from the University of Minnesota, who I mentioned to you had really taught me about turbulent mixing, Rodney Fox from Iowa State, who is a master in computational fluid mechanics, and his simulations really helped us design the mixing elements we use. Tom Hoy, University of Minnesota, is a chemist. I found out that Tom is a fly fisherman, as I am, and so we've become fishing buddies. Unfortunately, COVID canceled our fly fishing trip to Alaska this summer, but we're on schedule for next summer. And then at Princeton, Thanos and Yanis Kavrakidis were involved in the center. So this brought these multiple skills to this problem of how do we make nanoparticles in a scalable and efficient way. So now the next stop in this story is making stable drug nanoparticles using conjugation. And this story, my mentor here is Lawrence Meyer, an amazing man who really taught me all the important issues in terms of nanoparticle delivery, drug delivery, how you follows really important problems rather than getting doing things which are only scientifically or academically interesting. And Walid Saad, who is now a professor at the American University in Lebanon, was the graduate student who worked on this. And here's the idea. That is, a prodrug is a drug to which you've linked something else to make it a prodrug. And pro means that linker breaks and it releases the drug. Everyone else in the field had been working on taking these drug molecules that are, let's say, that are hydrophobic and linking them to something which was hydrophilic to try to make them more soluble. and to make them longer circulating. Lawrence's idea and our idea was, let's link the drug to something hydrophobic rather than hydrophilic. And that hydrophobic drug conjugate would now precipitate in the core of our particles. The release of the drug is determined by the hydrolysis or the linker chemistry that breaks that and releases the drug. This allows you to do two things. It allows you to control the release of the drug by not its intrinsic solubility, but by the chemistry of this linker. It also allows you to do two drugs and to have a drug cocktail delivered from a single nanoparticle. And here is some data from that work with Lawrence and Walid. This is the drug in circulation in a rat model. And by tuning this linker, we can tune the clearance time, or how long this drug circulates, from a half time of a day to just a few minutes, depending upon the chemistry of the linker and the chemistry of the anchor that we used. And this drug happens to be paclitaxel, a cancer drug. So this worked wonderfully well. We have patents on this with Lawrence, with Celator. The company has gone on to be tremendously successful. It was purchased by Jazz Pharmaceutical for $1.4 billion about a half dozen years ago. I had no commercial interest, so I'm still a teacher. Lawrence took his money, built a beautiful chalet in Whistler, British Columbia, where Celator is located, and spent 360 days on the ski slopes his first year in retirement. But to Lawrence, I owe a real appreciation for what's really important in nanomedicine. The next story is encapsulation of proteins and polypeptides. And this starts with Paul Burke, who is probably the most capable leader or guider in developing people's careers. He's an amazing scientist, but an amazing person in developing people who work for him and under him. And Paul was at Amgen. He had heard about our work, and he approached me and said, do you think your technique will work for encapsulating proteins and polypeptides? And the people involved, Suzanne Diato was a graduate student, and Marion Gindi was a graduate student who was working with me and with Thanos at the time. So this story technically is going to involve Paul and Thanos and Suzanne and Marion. I put Pablo here in parentheses because I just want to make the point that the story I'm going to tell you is one in which simulations play a key role in understanding microscopic behavior you can't experimentally visualize. And with Pablo, we began to have these wonderful collaborations which Princeton enables because the boundaries between people are so small. We're such an intimate size. And so Pablo and I have two papers with different sets of our students published in the last year. And so this collaboration between my interest as an experimentalist, the simulation interest of Thanos, Pablo, Giannis, have been really instrumental in us making progress. So here's the story. Paul gives us these series of peptides and says, can you make nanoparticles out of them? Some of them that were quite hydrophobic beautifully make nanoparticles. It makes a 100 nanometer particle. It's stable for 12 hours. It works perfectly. However, there were some surprises on some of the other peptide sequences they gave us that required Thanos and his students to go in and do these Brownian dynamic simulations. What's shown in this simulation is the purple is the drug, and the green here is the polymer stabilizer that we want to put on the surface of the nanoparticle. Here were the surprises that we got. So for this particular peptide, which is called bombesin, it's a therapeutic peptide, immediately you form these 30 nanometer particles, but within 30 minutes, it popped over to be a much bigger 800 nanometer particle. And it's not that it was unstable and kept growing forever, it stopped at this size. And so we said, why is this happening? And that's where the simulations really demonstrated the phenomena occurring. So those peptides that we're using, if they were very hydrophobic, they made a hydrophobic surface, and those hydrophobic blocks of the polymer landed on the surface and stuck. And you make small particles as shown in this quadrant, this box. If the peptide aggregated, but it wasn't very hydrophobic. Then the hydrophobic block copolymers like themselves more. They made micelles, which I described to you earlier. When they landed on the surface of the nanoparticle, they clustered. And so you left unprotected parts of that nanoparticle, that 30 nanometer particle. It would then aggregate over time until you had a now dense enough layer of these stabilizing polymers to stop further growth. But it first made this small particle, and then those later more slowly aggregated to form the big particle. So here was a case where simulation really helped us understand the detailed molecular mechanisms in a way that we couldn't by any other way. Next story is wonderful collaborations with my friend Rodney Priestley, a tremendous colleague, and again it involves simulations with Thanos. And the problem is making Janus nanoparticles, or biphasic two-phase particles. And what are these? This has been a very hot area, trying to make nanoparticles, small particles, which have two different compartments in them, or two faces. It comes up with the Roman god Janus, who's a two-faced god who you might have seen back when you studied Greek and Roman mythology. But Rodney and I had two students who were just tinkering on their own, not because we told them to, and we're working on precipitating polymers into nanoparticles. And then Rodney's real research interest is in how confinement changes what polymers do in confinement. And previously, he had worked on very thin films, 100 nanometer or thinner films. But the question was, if you put those into a spherical nanoparticle, could you see interesting confinement effects? And so Rodney and I started working together in work that he led. And sure enough, you can put two different polymers. They will phase separate once they form a nanoparticle and make these Janus or biphasic particles. And you can vary the ratio of the two polymers and vary the ratio of those two faces. The simulation part here has been, this is the simplest result of this research. When you make a particle with two faces, you can also make much more complicated geometries. And those again were geometries that Thanos's students doing simulations were able to understand and display how you can get even more complex geometry when you have this phase separation in confined nanoparticle form. So that's the second story, again, of collaboration and using the technique. Now we come back to siRNA for gene silencing. And Paul Burke comes back into the story. And Marion Gindi is a key part of this story, and Varun Kumar is a graduate student. So this story evolves that Paul Merck has just purchased Sirna. It's an enormously expensive purchase of this siRNA technology. Once they got it in-house, it became Merck's largest single research project to understand how to develop this Sirna technology, silencing RNA technology. Marion Gindi was also sent back to get her PhD at Princeton by Merck, and had worked with, I said, myself and Thanos. She's now back at Merck in Paul's group. They realize that they don't have a technique to make these nanoparticles in scalable form. So they contact me, and I said, let's see if we can do it. And Varun Kumar was a really gifted grad student who did this. In this story, what we have is a small siRNA fragment, which is very negatively charged. And what you do is you couple it with a cationic and oppositely charged lipid, which is hydrophobic. We had no idea this would work. That is, if in our rapid precipitation, because the siRNA comes in an aqueous phase, this comes in an organic phase, if the electrostatics would be fast enough to make it hydrophobic so we could cluster it. But sure enough, it works beautifully. We can make these 80 nanometer particles that are scalable RNA encapsulated materials, and it works for RNA and mRNA. We again had wonderful publications with Merck, as well as a beautiful patent. At this point, Varun finishes. Of course, Merck wants to hire him, but he decides Merck is too big a company. He wants to go with a smaller, more nimble company. He goes with another siRNA company that's a big competitor to Merck, which is a little awkward for me, but the students get to do what they want to do with their careers. Soon after that, Merck exits siRNA. and gives all their technology, including these patents, to that company that Varun now works for, which was kind of funny. And then through another sequence of a little more complicated history, those patents now reside with the largest mRNA company around. They're the basis for all of their production of their therapeutics and the COVID vaccines, which we all hope to see very quickly. And this has been just tremendously satisfying for me, is to see our technology develop into the main production mode for these mRNA vaccines and therapeutics. And so it's just been a wonderful reward for having, again, wonderful students to work on these problems. At this point, the talk diverges into two completely different paths or directions. And what I'm first going to describe for you is the Gates Foundation and global health. So the title I put, High Tech to Global Health. Everything I've talked to you about so far has been to make these beautifully designed nanoparticles using block copolymers to deliver drugs by usually IV injection. The Gates Foundation is an amazing organization, and they funded lots of research, and they realized that they were funding the development of brand new molecules, but then there was no way to get those new molecules into production, into clinical trials. And so they hired a new group inside Gates, and one of the main people there is Nia Bowers at the Gates Foundation, to find these paths to make these newly discovered drugs available for global health. Now, there's some really different requirements here than we had been worked with in the past. For example, everything has to be oral delivery. These are not injectable drugs. They have to be tablets that you give to people in these harsh sub-Saharan environments. It needs to be really low cost. The block copolymers we've been talking about are a couple of $100 a gram, and that's way out of the cost realm where you can have a drug that works for global health. And all of these drugs that we're talking about are very hydrophobic, and therefore are a problem to deliver. And then the important thing, it needs to be translatable and scalable. So the Gates Foundation approached us and said, would our technique work? They had looked at lots of techniques and decided ours is the one they ought to pursue. And we began this wonderful collaboration sponsorship by the Gates Foundation. Here is part of the team who's worked on this, and some new people have come into this. So as I said, we've now worked on 6 drugs. 5 of them we can successfully make into nanoparticle form. These are drugs that are very hydrophobic, and when you take them orally, not much gets into your system. This is the first one we worked with, which is called clofazimine. It's for a bacterial infection called Cryptosporidium, which causes diarrhea in infants and leads to significant mortality in underdeveloped countries in global health. And but instead of these really expensive block copolymers, we had to see if we could make these using things that are very inexpensive. So we started by looking at a cellulose, that's just wood fiber, a polymer called HPMCAS. Lecithin, that's PAM that you spray on your frying pans to keep food from sticking. And there's a zine, which is a corn protein, which is used to keep corn flakes crispy in your milk. Okay, so these are all really low cost materials, which are amphiphilic, which means part of the molecule likes to stick on hydrophobic surfaces, part likes being out in water. And sure enough, it worked well. It was a lot of hard work, but it worked very well. And this shows why we're trying to do this. This is the concentration that's dissolved. This is if you take the clofazimine drug crystals as you receive them, and you put it in this experiment over 30 minutes, which represents gastric fluid exposure, and you get essentially no solubility. When you take our nanoparticles and you put them in, you get 40 times higher dissolution kinetics. That is, you're dissolving a lot more of it, it becomes much more bioavailable and gets through your GI tract into your bloodstream. Why does it become more bioavailable? That's a sort of a technical discussion. It has to do with the drug not being in a crystalline form, but being in an amorphous form, a disordered form, and that makes it more soluble. Well, it's nice to have these results that we're able to increase its dissolution kinetics, its bioavailability, but you've got to do that in a cost-effective way. Our precipitation is cost-effective, but you have to end up with a dry powder. And so everything we'd done earlier had been for lyophilized materials. That means you freeze and then dry them, that you can do for expensive oncology drugs. But for the Gates Foundation, you needed to spray dry things. That is, you need to spray them just like you do powdered milk. And so in fact, we were able to take our original nanoparticles, spray dry them, redisperse them, and disperse them right back to the original nanoparticle form. So that was highly successful. I'm not going to go detail on this, but Gates wanted this scaled up. And let me go back to this picture I didn't mention. When we began this project, the Gates Foundation said, okay, we want you to develop this technology, but then translate it to a manufacturing facility that can do this at scale and will make the drugs. And that manufacturing facility is Wuxi Aptech in China, in Shanghai. And Nia said, you need to contact the head of formulations at Wuxi Aptech, who is Sunny Panmai. In this very small world way, Sunny was my PhD student about 30 years ago, who had his career in Merck, and then 2 years previously had left Merck and was the head of all formulations at Wuxi. So we've had bi-weekly phone calls translating all of this complicated geometry, and we're doing this manufacturing or production in our lab at 30 liter scales, which is much larger than I ever expected to do in our laboratory. And Wuxi is doing it at 300 liter and up to 100,000 kilograms per year. And at all these scales, as I mentioned, this scalable, the fact that we can work in the lab at milligram scales and make a nanoparticle, we can do syringe pumps, and we can do this hundreds of liters a minute scale and make exactly the same nanoparticle. And that's been the real triumph in this work. So that's taking a drug that's very hydrophobic, making it very small so it dissolves quickly. And here's the other branch of this, which I said is in an entirely different direction, and that is inverse flash nanoprecipitation. And this story involves this person right here, Shahram Hijazi. Shahram, even though in this picture just has one head, he actually wears 3 hats, and he wears them very well. So one of these hats is he's a senior person in a venture capital group. The other hat is he teaches 2 of the most successful courses in entrepreneurship here at the university that the students are just wild about. And the third is that he and I were talking about our technology. He had led Kodak's imaging business before he left to go in venture capital. And he said, let's take your technology and make diagnostic particles for imaging. But then after that discussion for a while, he decided that the part of the pharmaceutical industry which is growing most quickly is biologics, soluble things like mRNA, peptides, proteins. And that's the direction we ought to go. Robbie Poggles in the center is a graduate student in my group. And Robbie said, let's see if we can turn around the flash nanoprecipitation technique. And instead of precipitating hydrophobic things out of water, let's precipitate hydrophilic things out of an organic solvent. And he tried it out and it worked beautifully. I'll just make another, So let's go to this without going into a lot of detail. What I just want you to see is we can make these 100 nanometer particles. And the important point here is that everything has 50% drug loading. And this was the challenge Merck gave us. They said, if you can do this and get 50% biologic loading in these nanoparticles, then it's really of interest because there's no other way to do this. And that's what Robbie was able to show for this wide class of materials, from small molecules all the way to peptides and proteins, that you can make these inverted particles at high loading, high encapsulation efficiency. And that's really where the Optimio story took off. This is, I think, probably the most exciting results, out of all the examples that Robbie showed. And that is, this is 50% mRNA in a 40 nanometer particle, and you make these beautiful particles. You have 95% encapsulation efficiency, and no one else can encapsulate RNA at these loadings and make these small particles. So this is a real, one of the highlights of the Optimio story. Optimios has now collaborations and sponsorships from 6 major pharma companies with different therapeutics made by this technique. So that's what I wanted to give you. It's really a walk through a series of thank yous to amazing colleagues and collaborators over a long period of time. Of course, it's all the grad students and postdocs who have really done the work. These are just snapshots from the various parties we have at our house, which has not happened during COVID time. I want to say that this network of friends and collaborators, it really takes a village, as Hillary Clinton has said. In addition to the technical side, I also want to just comment on the Princeton culture and infrastructure here that makes this possible. And a lot of that is administration too. So I really thank Pablo and the Dean of Research Office, who has really driven this. When I began here 40 years ago, innovation and entrepreneurship was, I'd say, tolerated. But clearly there's been an evolution in that. So today it's strongly supported and aggressively supported by Princeton, for which I'm very thankful. And in that administrative roles, I'd also thank Colleen Burris and her office, which connects us with outside corporate and foundations. And then my special thanks go to John Ritter, who's one of my real heroes inside Princeton. So John is the head of the IP and office, and Lori Zodikoff, who works with our patents and collaborations. As you can imagine, the negotiations with outside companies, pharma companies, becomes challenging. And John is just amazing and flexible and has done a wonderful job in supporting us and enabling these collaborations that have enabled this technology to go forward. So I want to thank you for that, and I'd be glad to answer any questions. Let me stop sharing and go back to the screen. Okay. Let's see, so I guess I'll read these questions. First one, you have both licensed technology to large pharma companies and you've licensed your technology to startups. Can you talk about what you think about when deciding which path to take? What are the pros and cons of each? Good question. The pros and cons of each is our research has always been collaborative. And so generally the groups we're collaborating with, whether it's a small company, startup like Optimios, or whether it's a big company, Merck, they're already there knowing why they want to collaborate. And when we get positive results, they're already there willing to sponsor those patents. Because of course, it costs a lot of money to fight for a patent. That original Brian Johnson patent took us over 10 years of fighting with the patent office. And John Ritter's office sponsored all of those patents and that fight for a long period of time, which we greatly appreciate. And so it's really who is there willing to do the battle with you and support you that determines where we go. It's not that I go around looking for people afterwards. You're already engaged with them during the invention process. What advice can you give academics who are thinking about starting a company from the technology? Find collaborators, people who really understand business, marketing, all those things that we don't as academics. So that's, I point to Lawrence Meyer with Cellator, leading that startup company early on. I point to Shahram Majazi. So I wouldn't try doing this without already finding those people who will be your guides through this process. I'm an academic, I'm not an entrepreneur, but I know who I want to get in contact with and to let me be guided by. So I think that's a crucial thing if you're doing startups. So successfully starting a company, it's, again, as I said, the leadership of that company. I've been involved in some startups where the students wanted to do the startup, but they put together a startup. But at that point, if you don't understand really what the market is, what the issues in getting to market and getting funding, those were not successful. So it's really that network of people to help you understand funding. Take Shahram Majazi's course if you're a student and want to learn that, and finding the right people to be your guides this whole process. What advice do you offer early career faculty interested in innovation and entrepreneurship? I guess my main word is do good science or engineering if you're an engineer, because in the Princeton context, it's not that entrepreneurship itself carries the day. It's the fact that you've done good science or engineering, which is the backbone of that. And in my time here at Princeton, I go back and I think the real leader in this was Steve Forrest, who was in electrical engineering, who did early work on organic light emitting diodes, which are now found in all our cell phones, for example. And Steve, as I said before that, Princeton had tolerated entrepreneurship, but sort of looked at it askance. It can't be really good science. And Steve was the one, I think, who really began this process of changing the culture by doing tremendous science and having a tremendous economic impact. And so do really good science, because that's what is required of being a Princeton faculty member. But then look for those partners who can help translate that science into things that may make a difference, to the world. So that's don't try to be an entrepreneur first and then do second-rate science and engineering. Any other questions here? I guess that's what we want to say. And so question about about the Princeton ecosystem has allowed you to foster so many collaborations with industry, develop new technologies, spin out new ventures. Part of that infrastructure I've mentioned has been the Dean for Research office, and especially on the negotiation side, John Ritter's office, and I said Lori Zodikoff, working with our patents and contracts. Many of my friends at other universities that are bigger than Princeton, let's say, have ossified procedures for interacting or contracting. And you just can't get around those procedures or those precedents or whatever. And the thing I appreciate about Princeton is we are a small place, and therefore the people can make decisions. We're flat enough organization can make decisions to do things that are maybe new and make a difference. For example, John Ritter's office, to make things work, to do research in the nation's service. And so the flexibility and our size, I think, is a real help in that process, and not having a big ossified, a rigidified structure or series of rules and regulations that keep you from doing innovative work. Then the other thing I'd mentioned, I think, is something that's happened in the last, let's say 10 years, is the number of new programs that sponsor relatively small grants, but let you try things without having to submit a big NSF or NH grant. Those have been occurring in the engineering school for quite a while. We've had sponsorship from the Helen Hunt Foundation, several others that allow you to do creative new things to see what you can do. And now Pablo has initiated many of those across the university. And those have been extremely helpful in letting us explore new things, letting us innovate in a way we wouldn't have been able to if we had to write an NSF or NIH grant before we tried any new idea. So that's, again, something Princeton should be really applauded for. And I'd also mention the new incubator space, the biolab space, which has been set up. I think there's more Princeton can do in helping small startups that are doing life science, material science, and I hope that more is done, but that's certainly an excellent first step in having that incubator space available. So Princeton's done a tremendous amount, especially in the last 5 or 10 year period, at supporting and encouraging the kind of innovation that I have a passion for. Any other questions?
Bob, I think we have time for one more question,
Okay.
if there is an additional question.
Yeah, let me make a comment towards postdocs and PhDs who are heading out. Should you go with a small innovative startup, or should you go for a big company? That is, if you're not heading to an academic career. And there I'd say, you really want to understand the small startup company, because most startup companies fail. And so you want to be bringing something unique to that company and understand the trajectory there. So there's a little more risk, but a little more potential upside in going to a small company. It's actually much easier to go from a big company to a small company. So, working with a major pharma company for a few years and then going to a startup is actually an easier route to get into some interesting startups. So the route you take, I think, going into startups from the beginning, you need to be sure you bring something that really has value and you understand the value proposition of the startup you're joining. Okay.
Great.
So thank you for this opportunity to share.
Bob, thank you so much. This was a really inspirational talk. It shows how one can translate really good fundamental science into technologies and products that make a difference in people's lives and in the world at large. And it's great to see how this happens in Princeton's unique collaborative environment. Thank you so much, Bob. And now I'm going to pass the baton back to Colleen.
Hi. Thank you again for sharing your experiences from your remarkable career and highlighting the importance of relationships. It's definitely important in the work we do in the Dean for Research Office. And thank you to all the attendees for coming together to honor Bob and celebrate this special event with us. Enjoy the rest of Engage 2020 and be well. Thank you.
Thank you, Colleen.